This is the SDS-PAGE Lab to separate proteins of cellular extracts, and we will be doing the two CRISPR-edited macrophage cells, and here we'll be using both a positive and a negative control, which the positive we will know, and this would be expressing the wild-type CCR5, and the one that would be a good positive control would be the unedited wild-type macrophage, because this has the undisrupted CCR5 gene, and the negative control will be the one that is the opposite of the positive and will show no result, hence no CCR5 protein, which means that the pancreas would be the example of the negative, since it has no, it doesn't show anything of the CCR5. Identifying the samples, the positive we know is the wild type cells, as mentioned. The pancreas is a negative, and our choice is the edit 7 cells. And the predicted outcome will be number 3, because in this image here, the protein has run all the way to the bottom for analysis. And then what's next after this will be testing the protein, because we cannot do any other tests after that. So here is the protocol: collect the cells, add the buffer, denature the protein, centrifuge the sample, load the gel, and conduct protein electrophoresis. So I gotta set the temperature to 95. Okay. Then I'm gonna follow the instructions. Set it to 30 microliters. Gotta attach a tip. And then I gotta close the box to prevent contamination. Now I gotta put it on a wild type cell culture and extract it. And then put it in the wild type cells. And I gotta do the same for all of them. So I'm gonna do that. Gonna put back the micropipette. And next is the P20 micropipette, which is this one. We gotta set it to 10 microliters. It's a bit sensitive. All right. Then we gotta attach the corresponding tip. Yep. Yeah, it was open. It was just a system. Now I gotta add the 4D SDS sample buffer and do the same as the previous one, but except I'll be adding the buffer, so I'll be doing that. Okay, no. What's the purpose of adding SDS? Stabilize membrane proteins, yep. Breaks down cell membranes and these, yep. it doesn't destroy proteins. Yeah, I'm sure it's all except C because all of them seem correct. Yeah, now I gotta add it to all of them as shown. Yeah, now I gotta repeat this process over and over for all of them. Put the microwave back. And now I'm gonna move my samples to the heat block over here. And then set it to 10 minutes. And push start. Now I gotta move all of my samples back here. And then open the microcentrifuge lid and move your 4 samples. Okay. I'm gonna, balance it. And then close it. Press the pause button. And then take them out. Now, place the polyacrylamide gel, which is this one, into the electrophoresis box. You gotta fill a cassette with buffer, the cassette. And then I gotta remove the comb. Then I gotta fill the outside with buffer. And now I have to take the micropipette, the P200, this one, gotta set it to 20 microliters now. Yep. Gotta touch a tip, corresponding tip. And then, sorry, I gotta move my micropipette so it's in the protein ladder tube, which is this one. And then I gotta drop the plunger. All right, close it. Move the micropipette so its tip is above the first lane in the protein gel. Here. Dispense the protein ladder by pressing down the plunger until to the first stop slowly. And now I gotta eject it into the biohazardous waste. Why should you micropipette samples, or how should you micropipette samples? Well, as I mentioned, slowly because it could spill, over the next walls. Yep. Now I gotta repeat the procedure. Gotta add the protein ladder. No, I gotta dispense 20 of the positive control, which the wild type cells, into the second well of the gel. All right. And I gotta do this for all of the other ones. All right. So we're gonna add some of this. All right. Put it here in the second one, and then dispense it slowly. Gotta repeat it for all of them. For the pancreas. And the third well. Slowly again. Yeah. Open the biohazardous waste. Dump the tip. Now it's gotta be the edit 4 and edit 7 cells. Into the fourth well. Slowly again. And the last one will be the edit 7 cells. Into the fifth well. All right. Eject the tip, close it, and now put the micropipette back where it belongs. Now, we gotta place the cover. Select the red and black cables and connect them to the supply power supply. All right, red with red, black with black. Yeah. Turn on the power supply. Turn it on. Set the voltage to 150. Set the timer for 60 minutes, which is here. All right, and then start the power supply. And then I gotta wait. Pop question, how do you know if the gel has run for long enough? I would know if it's way at the bottom of the gel. Yep. Now I gotta turn off the power switch and unplug the electrodes from the power supply. And then carefully remove the cover to inspect. Now, after it freezes, inspect your gel and compare it to the predicted results, actual results, and the ideal results. So this is what I predicted, and this is what I got. Well, they seem to be almost the same. The same. And the ideal results was very, very similar actually. Well, they are similar, as you can see. Yeah. And now we're gonna see the controls of what I predicted. I predicted the wild type for the positive, the pancreas for the negative, and the edit 7 cells because it was preference. And the correct was the wild type, yep, for the positive, the pancreas for the negative, and the edit 4 and 7 cells. That's right. Seems about right. Yep. And then the Kuma staining was with the blue one, which is here the CCR5. So here's the reflection. Let's look at what you learned. Just click on your answer with the following questions. Protein sample is separated using following technique. The technique we use is SDS-PAGE as it's known. Using SDS imparts an overall, it is a negative charge on our proteins. In a PAGE gel, proteins are separated by their molecular weight in this case. I received the following gel after electrophoresis. What went wrong? Well, it didn't run all the way to the bottom, so I'm assuming it's not time. You did not allow for enough time. Yep. After running a protein gel, you can inspect the gel for protein bonds and determine if your previous gene edit stopped working. Is this statement true? No, you have to go further and Western blot it in order to visualize your protein bonds. How confident do you feel about using the technique you learned? I feel very confident. And that's it for this lab. Thank you. I'm gonna read the summary now.